Xenotransplantation is the process of transferring living cells, tissues, or organs from one species to another. These transferred cells, tissues, or organs are called xenografts or xenotransplants. The development of xenotransplantation is, in part, driven by the fact that the demand for human organs for clinical transplantation far exceeds the supply. Moreover, recent evidence has suggested that transplantation of cells and tissues may be therapeutic for certain diseases such as neurodegenerative disorders and diabetes, where, again, human materials are not usually available. This article pertains to the FDA Forum. It was published as of the 28th of March 2019. The idea behind xenotransplantation dates back to 1667, where a French doctor named Jean-Baptiste Denis taped the veins of farm animals to perform human blood transfusions. It is only until the 1950s that surgeons practiced xenotransplantation on human patients. Xenotransplantation could benefit thousands of people by providing an unlimited supply of cells, tissues, and organs with many uses, which include organ transplants, cell transplants, tissue transplants, and bridging transplants. The immune response to xenotransplantation is initiated by a number of components of inflammation and immunity. The major problem, however, of xenotransplantation is the rejection of porcine cells, tissues, and organs, which occur mainly as a result of cellular and hyperacute rejection. Recent studies have also revealed the involvement of macrophages, which act like immune cells in host defense in the immune rejection of organ transplants as well. Physiological compatibility cannot be assured. A review of the physiological performance of clinical cases of renal, cardiac, and hepatic concordant xenotransplantation demonstrates some incompatibility between even closely related species. You may ask, how then can scientists allow cells in the immune system to accept xenografts from a completely different species? Well, the antibodies characterized most extensively in recent years of xenoreactive antibodies, typically defined as those antibodies which bind to the cells of a foreign species. Xenoreactive antibodies initiate the hyperacute rejection of xenografts and help minimize cellular or hyperacute rejection. The above points are further detailed later in the documentary. Pigs have been identified as potential donors, primarily because of their physiology and anatomy being similar to humans. Moreover, there are no major ethical considerations regarding their use in medicine. Finally, their potential has been increased by the application of the most recent techniques of genetic manipulation, which is transgenesis and genes transfer, through viral vector and cloning, through nuclear transfer. However, some significant problems remain regarding their use: immunological barriers and risk of viral transmission. Scientists have found a solution to these problems by manipulating the pig genome, targeting the human immune system. One of the strategies to prevent rejection and viral transmission of xenotransplantation is the production of pigs no longer expressing the major xenoantigen alpha-gal. This is done by genetically altering the genome with human DNA and pig DNA to reduce the expression of alpha-gal as much as possible, to remove as much risk. There will always be a risk of mutation and rejection, but this method significantly lowers the risk. The implementation of xenotransplantation in surgery is significant because it could potentially provide an unlimited supply of cells, tissues, and organs for humans. Any disease that is treated by human-to-human transplantation could potentially be treated by xenotransplantation. Organ xenotransplants could include whole hearts, lungs, livers, kidneys, or even pancreases. Tissue xenotransplants could include skin grafts for burn patients, or corneal transplants for the visually impaired, or even bone transplants for those needed limb construction. Cellular xenotransplants may provide treatment for people with diabetes, Alzheimer's, or even Parkinson's disease. Stephanie Faye Beauclerc, better known as Baby Faye, was an American infant born in 1984 with a hypoplastic left heart syndrome. She became the first infant subject of a xenotransplant procedure and the first successful infant heart transplant, receiving the heart of a baboon.
Due to the nature of xenotransplants, there aren't many people who have had this procedure enacted on them. However, what we do know is that this is viable for people of all ages. A newborn baby lived with a baboon heart for 20 days. There are also multiple ethical issues with xenotransplantations, and these include the tough questions of how will the creation and care of such donor animals be managed? Is it even morally right to breed an animal just to die for a single specific organ or organs? What are the emotional impacts on the recipients of such donations? And what's the fairest way of allocating these animal organs? Even when questions like these are answered, it is likely that xenotransplantation will still be a subject full of controversy. Despite all this, there are multiple major obstacles when it comes to xenotransplantations, and these are mainly severe immunologic barriers and physiological differences. These immunologic barriers include hyperacute rejections, acute vascular rejection, cellular rejection, and chronic rejection. In hyperacute rejections, antibodies, proteins that bind to foreign invaders and tell the immune system to get to work, bind to the donor's endothelium, which is the interior of blood vessels, causing inflammation, thrombosis, the formation of a blood clot, and necrosis, the death of cells of the transplanted organ. Acute vascular rejection is a multitude of more times, however, more complex than hyperacute organs, and is therefore not fully understood yet. However, what is known is that this type of rejection is driven by interactions between the donor organ and the recipient's antibodies, macrophages, cells involved in the detection and destruction of harmful organisms, and platelets, blood cells that form clots. The resulting response includes intravascular thrombosis and necrosis of vessel walls. While hyperacute and acute vascular rejections are based on the immune system's response, cellular rejection is based on cellular immunity, wherein natural killer cells attack and damage the xenograft. Chronic rejection, the last of the immunological barriers, is slow moving and progressive, and usually occurs in transplants that have survived through the initial rejection phases. Scientists are still not sure how chronic rejection works, but it is thought that the major cause of chronic rejection is when lymphocytes, immune cells, activated by antigens in the vessel wall of the donor organ, activate macrophages to output smooth muscle growth factors, resulting in a buildup of muscle cells on the vessel walls, which then causes hardening and narrowing of the vessels within the donor organ. The issues in physiology will include the size differences in organs, as well as the lifespan of said organs. Pigs only live to 15 years, so would a xenograft from a pig last longer than that? There are also hormone and protein differences, as well as differences in the environment that the organ resides in. Three questions that I still have would be 1. Can xenotransplantation be done the other way around? For example, human organs are put into animals. Does xenotransplantation of limbs work? And can xenotransplantation be used to combat diseases?